Okay, can you all hear me now? So on behalf of Ophelia's tenure committee, I want to welcome you all to her talk. And I also want to say it has been an honor to be on her tenure committee and to look at her progress. So Ophelia started out as a undergraduate at Stanford. She majored in biology and math, minored in math, and she's maintained that balance between quantitative science and biological science throughout her whole career. And so then she went on to Caltech for her PhD, and her postdoc was at Berkeley before coming here. And she's tackling probably one of the hardest problems in biology, which is the microbiome, incredibly complex. And it's not just in, our gut microbiome, but a lot of the work that Ophelia is doing has broad applications to other microbiomes, like soil microbiomes, for example, or there are microbiomes in almost every sphere of ecology that you can think of, many, many different microbiomes. And so her approach is using basically the principles of chemical engineering, which are mathematical modeling, looking at fluxes in and out of systems, looking at developing dynamic models with the intention of being able to make predictions about how microorganisms will behave in exceedingly complex situations like the gut microbiome. And so she's really done a tremendous amount to create an incredibly dynamic research program. I want to just list you some of the awards that she's gotten for her work. She was one of 2 winners out of 350 disclosures for the WARF Innovation Award back in 2019. She was featured in Biochemistry as Future of Biochemistry. She received an NIH Outstanding Investigator Award, a MIRA Award in 2017. She was an Army Research Officer Young Investigator. She received the Shaw Scientist Award from the Milwaukee Foundation. And she also, prior to that, received a Life Sciences Foundation Postdoctoral Award. So the other thing that I want to mention is something about teaching, and that is that Ophelia and Phil together have put together a very impressive high-level undergraduate course in modeling of biological systems. And I've attended that course a few times, not the whole course, but parts of it. And I was incredibly impressed with the level that she's been able to pull the students to in terms of their own writing Python code and modeling of the biological systems. Very effective teaching and very effective in elevating the students to a level that probably they themselves didn't imagine they would be achieving. And I think that's the mark of really great teaching. And I actually asked Ophelia if she would just spend 1 minute toward the end of her seminar talking about what she has accomplished in that class. I don't want her to be too modest. I think she should talk a little bit about what she's done in the class. So her seminar is entitled Predicting and Designing the Human Gut Microbiome.
All right, everyone can hear me. Thanks, Alan, for the really nice introduction. It's been a real pleasure to be a part of the biochemistry department, and I'm excited to tell you about some of the work that we are doing in our lab. And Alan gave a nice introduction on the importance of microbiome, so maybe I don't need to actually spend time motivating it more broadly. But before we get started, I wanted to thank current and past members of our research group. And in particular, members that started the lab from basically nothing, an empty room with no anaerobic chambers. And in particular, that's Susan and Ryan Sue and Ryan Clark and Josh Hamilton and Yu and Sonali and James, they were really the ones that like started in the lab with not much to build on, and they really created incredible resources and also supported many new students and postdocs that have joined the lab and really created a very, very important foundation for our group. But our group is diverse both in terms of like scientific expertise, spanning engineering, microbiology, biochemistry, mathematics, but also in terms of the perspectives of the people that are in the group. And that really has been very critical for our cross-disciplinary research program. So we're really thinking about complex problems, and I think that diversity of perspective has been really enabling and powerful and critical. So wanted to thank our group. Our gut microbiome contains trillions of bacteria, and they have co-evolved in our gut over thousands of years. And there's a delicate balance between beneficial functions that those bacteria are doing to enhance our health and provide nutrition for us, but also dysbiosis. So under certain types of environmental cues, the microbiome can enter a state where it causes and reinforces disease. So we have, over the past decades, really mapped the microbiome composition. We've looked across different individuals in different disease states, healthy states, and sequenced the communities with next generation sequencing. So that allows us to understand the kind of expansion and contraction of different types of taxa in the gut and how they're linked to different human diseases and also influencing our physiology, our behavior. And so this has been really important for providing a foundation for understanding what these microbes could be doing. But there's a lot of unexplored, unanswered questions that we cannot get just by looking at the microbiome under these different states. So these observational studies. I think they've provided a foundation, generated hypotheses, but we really need to go much further to understand what the microbes are doing, how the communities assemble, what makes them stable, how do they respond to perturbations? Can we predict their response to perturbations? Can we tailor metabolic outputs to our benefit? And so those things are really lacking right now in understanding the microbiome. And it's an incredibly complex system. There's also been another remarkable discovery, which is been known for a long time, which is if you take stool from a healthy donor and transplant it into a patient that has recurrent C. difficile infection, that this can actually cure the patient with this recurrent infection more effectively than antibiotics can. So that already suggests that there's something remarkable about gut bacteria. They can actually treat human disease. But this has not been solved for a wide range of human diseases, so there's a lot of work we have to do to understand how to tailor these gut bacterial communities to our benefit and make them effective. But also, fecal microbiota transplantation has many disadvantages and limitations, including the fact that it's not standardized, could be transferring antibiotic resistance pathogens, for example. It's not scalable. If we find good donors, how do we scale that material? It's really challenging. There's so much variation. And then also, we cannot easily tailor it. So if we wanted to tailor or personalize these therapies, how do we do that? We just have a mixture of different bacteria that are incompletely characterized. So this provides kind of motivation for studying the microbiome, but really is not a solution moving forward. So moving towards a more mechanistic understanding would allow us to be able to tailor these microbiome functions. And over the past decade, there's been many metabolites and compounds that are released by bacteria in the gut that have been linked to human health and disease, mechanistically. And some of these include degradation of dietary fibers, for example, into fermentation end products, which are produced in millimolar quantities in our gut, and have many different effects on human health, notably butyrate, which is a very beneficial metabolite. And there are many other transformations, secondary bile acid transformations, which there's about 50 or 70 different types of transformations that bacteria can make to bile acids. And they interact with human cells and our physiology, and they have many effects. All of these metabolites have been shown to impact different host physiological outputs, whether that's energy, harvest, cardiovascular disease, insulin resistance, or even our behavior. So these metabolites are being produced and degraded by gut bacteria, and these are specialized bacteria that do all of these transformations. But there's also bidirectional interactions, so these metabolites impact the gut community as well. Butyrate, for example, has been shown to inhibit the growth of certain types of bacteria. And then also there is a feedback loop between the host and the microbiome. So as the microbiome alters its environment, the host reacts to those changes, and that leads to these feedback loops, which can be particularly deleterious in the case of dysbiosis, where these feedback loops can actually reinforce inflammation. So it's a very complex relationship between the microbiome and the host. And our ultimate goal is really starting to understand these metabolites that are produced by gut bacteria and manipulating them. So for example, say we wanted to treat cardiovascular disease with a bacterial therapeutic. We would, if we knew which metabolites were involved, and in this case we have 3 at least that have been shown to mechanistically influence cardiovascular disease, we could increase butyrate, decrease trimethylamine, and decrease imidazole propionate. So if we can design ways to perturb the microbiome to shift these metabolite profiles, we could start having an impact on disease states such as cardiovascular disease. So we would like to understand how to design these microbiome interventions, but we are kind of confronted with complexity. So why is this not solved yet? We've already identified a lot of metabolites, but why can't we actually go in and manipulate these metabolites to our benefit? So there are hundreds of species in the microbiome. They're unknown and complex interactions. These could be between different pairs of bacteria, or it could be between 3 bacteria, where the interaction between pairs is impacted by a third member. This is an example of a higher order interaction. There's temporal variation, spatial heterogeneity, single cell phenotypic heterogeneity. Even with an individual species, we find that 50% of a given strain contains a polysaccharide utilization loci. That suggests that even at the strain level, we have a lot of variation. And also, microbiomes are kind of a physical system that has collective behaviors. And we can think about these collective behaviors as a phase diagram. This was a nice study that was just recently published from Jeff Gore's group that showed that the system, microbial communities, can have different types of qualitative behaviors. Qualitative behaviors meaning either species coexist, they go extinct, or they have stability, or different degrees of stability, just as a function of different types of parameters in the system. In this case, this is diversity, initial diversity of the community, and interaction strength. So it's a phase diagram. We can change the behavior of the system, but we don't really know how these systems behave as a function of different inputs. And the kind of the collective behavior of the system depends on the interactions between the parts. So that's why this is not a solved problem. And that's where we are. And so we're interested in taking a different approach to studying the microbiome. And we're not alone. There's many other groups that are studying this as well. But we're interested in a bottom-up approach to microbiome. So a bottom-up approach allows us to control the initial concentrations of different taxa or species in our system. We can vary either the presence and absence of different community members, represented by this gray and white, or we could vary the concentrations over a wide kind of continuous range. But that gives us control over the species abundance, which is actually a really powerful way to understand how interactions and species are contributing to a system behavior. And so this creates a kind of combinatorial explosion. So just for 25 species, we have 33 million possible combinations of different species. And then if we take the number of species roughly in the human gut, that gives us 10 to the 30 possible combinations of different communities that we could create. We can't do that, obviously. So we can't build these communities in the lab at this scale. So we have to turn to other types of approaches, such as computational models, that allow us to really navigate what we refer to as a structure-function landscape. I know others have different definitions of what a structure-function landscape is. But talking to a biochemistry group. But in our case, our structure function landscape is mapping the initial abundance of different taxa to a function of interest, let's say butyrate concentration. And so this landscape could be rugged with many peaks and valleys. And so we want to identify how to navigate this really complex landscape. And we can exploit tools from engineering and computational modeling and optimization. So I've kind of introduced a little bit about this bottom-up approach. We have this toolbox of different microbes. We build communities in the lab using different types of experimental platforms, ranging from very low throughput experiments in germ-free mice to very high throughput experiments, million or 10 to the 7, different communities that we could build in these microfluidic droplets. And so we can combine these data sets with computational models really to explore this very complex and rugged landscape to understand microbiomes at a more mechanistic level. So I'm going to tell kind of 3 vignettes today. I would love to talk about more, but there's time limits. And so I'm going to highlight kind of just 3 projects that we've been working on over the past few years. So the first project will discuss community metabolism in the gut and also touch on how we can enhance stability and robustness in gut communities. So a very talented postdoc in the lab, Dr. Ryan Clark, was excited to take a 25-member synthetic human gut microbiome, which contains, it kind of represents a healthy human gut. There are 5 butyrate-producing bacteria in this community. Butyrate is a specialized function. So the pathway is only present in a select group of species. And the other species represent diverse phyla, spanning all the phyla in the gut, and also having important metabolic roles in the community. Many of these bacteria are highly abundant across the human population, highly prevalent, and also have links to human health and disease. So that's how we chose our community. We also chose it specifically to study butyrate production. So depending on the question, we can design different types of communities to study different types of metabolic functions. And so we took an approach where we have the ability to control the presence, absence of different species in the community, and we can combine these data with computational models that allow us to not exhaustively explore this 33 million possible combinations of consortia so that we can understand how interactions shape butyrate production. And I should mention that butyrate producers have a very unique set of niches in the gut, and so their interactions with other gut bacteria are really important for us to understand. We also had a great collaboration with Al Hero's group and Mayak at the University of Michigan to really start to explore new types of models for studying the microbiome. So we took an engineering-inspired approach where we use something called a design, test, learn cycle. This is frequently used in engineering fields, and I think now being more commonly used in like synthetic biology and engineering, where we take some initial data in the lab that we feel is informative based on maybe underlying hypotheses or knowledge about the system. And we use that to build some communities, take some measurements, and then we build a model with that information. And that model can be used to guide subsequent design, test, learn cycles. So it's an iterative process and allows us to efficiently explore a really complex system. And so in our case, we started off with single organisms and all pairs with butyrate producers. So we have 20 species that don't produce butyrate, and we have 5 species that do produce butyrate. So we can look at all pairwise communities of non-butyrate producers with a given butyrate producer or butyrate producer combinations. So we use that data to build a model, a computational model, which we can use to simulate all possible communities. And we use that model to design a set of lower complexity communities, 3 to 5 species, because we knew we were lacking some information, so we didn't want to jump to a very large system without kind of learning a bit more about the types of interactions that were happening in the system. And then we used this model to design the communities, tested them, and we updated our model with this new information. And so that might look something like this, where we have a model that has maybe a moderate predictive ability, and then we can use the model to learn from the experimental data and then improve our model's predictive capability substantially. And so we use this to design a set of 10-member or communities of at least 10 members. And then we use this system to actually validate our model's predictive capabilities. So we validated along the way and found that the model really needed information from these really complex communities to be highly predictive of other complex communities. And so we can take this trained model that was trained on about 415 different communities, and we can use that to now predict the metabolite profiles and composition of new communities the model's never seen before. And so this is just kind of a summary of our overall kind of workflow here, and our interest was actually focusing on these fermentation end products, in particular butyrate, but we also have acetate, succinate, and lactate. And each of these molecules are produced at very high concentrations in our gut and vary across individuals. And so we'd like to be able to tailor these metabolite profiles to observe their effects both in vitro and in vivo. So this is, I'm going to show some data here, but I just wanted to start off showing kind of the relationship between butyrate and lactate. So these are just, there's 4 outputs to our system, but we can't visualize in 4 dimensions. So we're looking at this in 2 dimensions, and we can see that the 25-member community, which is all species mixed together, trends toward kind of maybe an intermediate, actually lower butyrate concentration and intermediate lactate concentration. So then we can use our model to simulate all possible communities. So now this is 26 million different communities, because it's all communities with 10 or greater species. So each data point on this plot represents a different community where we varied the presence-absence, and we can predict the relationships between different metabolites in our system. And we can see that there is a bimodal distribution that's predicted here. There's a bacteria called Acaciae, which is a very prevalent butyrate producer that uses lactate to transform into butyrate as one of its modes. And we would expect a negative correlation between these 2 variables because lactate is converted into butyrate. So the model, with about 400 different observations, was able to predict these relationships between metabolites that's really governed by the underlying metabolic network of the system. And so we decided to take this model and test to see if the model could design communities with extreme metabolite profiles. So these are metabolite profiles that either minimize or maximize all of 4 combinations of metabolites. And so what we call these communities is called corner cases. They're just the extremes of these distributions. And so we can go and either randomly sample communities as another set, or design these extreme communities that have maximize or minimize all 4 combinations of metabolites. And the model's really challenged because there are not a lot of examples in these sparse regions of the design space, whereas there's many more points in these more dense regions. So the model's really challenged to predict the extremes. And so here is the predictive kind of performance of the model. So this here on this axis is the cross-validation accuracy. So it just measures the goodness of performance of the model. So a value near 1 means perfect. The model is extremely good. And then a value near 0 means that it's not very good. And so we see that for these 4 metabolites, butyrate, acetate, lactate, and succinate, they have a high cross-validation accuracy above kind of this dashed line. And then we can predict also the species as well, which there are 25 species, so that's 29 different variables in our system. And overall, nearly every species is very well predicted, showing that our model can kind of capture and predict the behavior of the system. And we can also benchmark our model against other models, which we actually built in the past, which is more mechanistic. So our new model is very flexible. It can capture really complex interactions between variables. But this this kind of machine learning model can capture the system behavior much more accurately than our mechanistic models for all 4 metabolites. So that shows that this model with limited information can be really powerful for predicting microbiome metabolite composition, metabolite concentrations and species abundances. But these types of very flexible machine learning models have one major drawback, which is that they're typically black box. So we don't really understand a lot about what's actually happening under the hood. But we have some tools that we can use called local interpretable model agnostic explanations. Essentially is looking at the sensitivity between inputs and outputs of our model. And so we can decipher how microbes impact metabolites and how microbes affect each other's growth. So for example, Acacia is one of the dominant butyrate producers in our community, and we see a very strong positive contribution towards butyrate, whereas consuming lactate to produce butyrate, so there's a negative relationship between Acacia and lactate. And then we also can look at kind of all of these interactions, and we found kind of confirmatory interactions where we know, Acacia produces butyrate, but we also discovered some new interactions in the system that we didn't know existed before. And so this allows us to really understand how microbes are shaping metabolite profiles and growth dynamics. And we can also identify contributions of either species or pairwise interactions towards these functions. For example, we found that succinate and acetate are driven by interactions between species, whereas butyrate and lactate are driven by single organisms. We also found that Bacteroides tend to really shape growth dynamics. They're really highly abundant in the gut and in these communities as well, but they don't really tailor metabolite profiles. So it's the other organisms, the firmicutes, proteobacteria, and the actinobacteria that are really specifically tailoring metabolite profiles. So we were able to take this model and design some communities that had really interesting functions. So here is experimental data. This is no longer model prediction, but on the x-axis we have the total amount of butyrate producers. So we have 5 butyrate producers, so we can just sum their abundance. And on the y-axis we have butyrate concentration. Down here we have the 25-member community. It's really suboptimal, not producing a lot of butyrate. The 5-member community, no competition with other gut bacteria, should be producing the most. It's kind of intermediate. And then we discovered these new communities up here that have very high levels of butyrate, and we're not changing the butyrate producers. The butyrate producers are present in all of these communities at the same initial abundance. The only thing that's changing here is the initial abundance of different non-butyrate producing bacteria. And so just to kind of illustrate this, we have the 25-member community, which produces kind of moderate levels of butyrate. And then we have these support communities. There are species that don't produce butyrate, but they're enhancing butyrate production substantially. So this shows that interactions between bacteria can be engineered to enhance desired metabolic functions. And we're applying these approaches in the lab towards other types of applications. We're looking at biological nitrogen fixation and engineering support communities to enhance plant growth. We're also looking at transforming waste into medium chain fatty acids and designing support communities that can enhance medium chain fatty acid production. So this general principle, I think, could be applied to a wide range of systems, health relevant and beyond. So with all of this modeling, what did we learn? So we learned biology. We learned about how different organisms interact with each other. We found some species that inhibit butyrate production through either resource competition or acidification of the environment, hydrogen sulfide production. We also found species that seem to support butyrate production by buffering the pH or providing substrates to butyrate producers to really enhance their funneled flux towards butyrate production in the system. And we also discovered kind of like this AND gate, which is interesting. We have either a pH buffering species and a strong competitor that together can enhance butyrate production. So this points to kind of higher order interactions. Interactions are not just pairwise, but they're occurring between combinations of species that really can lead to kind of emergent effects in these multi-species consortia. So we were really excited by this, and we were interested in seeing whether our results could potentially translate to a new environment. We had some communities that we optimized, we designed and tested in a single media that had high butyrate production. And this is kind of another simulation plot. We're using our machine learning model to predict the butyrate concentration and butyrate producer abundance for all possible communities with 10 or greater species. There's one bacteria called D. piger that inhibits butyrate production through hydrogen sulfide, which we kind of tested independently and found this interesting relationship between hydrogen sulfide and butyrate production. So that's where the red points are. They're kind of trending towards lower butyrate production. And so we identified these two communities, what we call COM5 and COM6. And in our experimental data, this is where they fall. So COM5 and COM6. This is the same axis here, except this is observed butyrate producer abundance and measured butyrate production. And we chose these two communities because they had high butyrate producer abundance. So we hypothesized they would be more robust. And also they have very high butyrate production. So we wanted to ask, can our model optimize communities predict butyrate in completely new environments, such as the mammalian gut? And so this experiment was led by Erin Ostrom in the lab, and she decided to take these communities and introduce them into germ-free mice. So these are mice that lack a microbiome, they're sterile. And so we can introduce defined consortia. It's really powerful for studying host microbe interactions. We can also define the diet, so we can control exactly what nutrients these mice are getting. And we have, as our reference, kind of negative control, this 25-member community. This is a community of mixture of all organisms. We expect this community to have low butyrate, and we expect these two communities to have high butyrate. And we also chose kind of an inulin diet, which is a carbohydrate composed of fructose. It's a pretty simple carbohydrate, but it can be easily introduced into the diet. Glucose, which is in our media, is very difficult to reach gut bacteria because the host absorbs a lot of glucose. So that's why we have to use fiber. And we also contrasted this with a cellulose starch diet because the bacteria don't have a lot of accessible carbohydrates from this diet. So this basically allows us to analyze the contribution of inulin to the effects that we're seeing. So we introduced these communities, we fed them kind of the different diets, and then we monitored community composition, and then we look at the short chain fatty acids at the end of the experiment after about 4 weeks. And this is kind of a heat map of the species presence and absence for these two model design communities, COM5 and COM6. And so we can see that that there's a fair number of species absent and also differences between COM5 and COM6, whereas this is a 25-member community containing all the different bacteria. And we find that about after a week, the communities stabilize to a steady state with differing numbers of different bacteria that are colonizing depending on the membership of the community and the diet. And then when we look at butyrate concentration, we were really excited to see that COM5 and COM6 are producing very high levels of butyrate. And just as a reference, kind of a conventional humanized mouse fed an inulin diet is usually around like 1.2. This was previously measured by Federico's Ray's lab. And then other defined communities are around 1 to 2, but our communities on average are producing much higher than that. There's a lot of variability. This is a new experiment that we just got the data for, so we're excited to repeat this again. And then also we can see the composition at the endpoint. But I think kind of the main point that I wanted to convey here is that we used a model to find these communities. If we had chosen a community on average, we would have chosen a community that trended towards lower butyrate production because the density of points is much higher towards low butyrate production. So there's kind of an attractor state where most communities are going to have low butyrate production. But we use the model to find these really interesting communities in these sparse regions of the design space. And then we use the model to design these communities, and then we tested them in mice. And now we find high butyrate production. So we're really excited about this. I think this shows and highlights the power of models to really identify these unique biological systems so that we can really start to dive more deeply into the mechanisms and use these as potential therapeutics. So now that we're talking about fiber, I wanted to mention a little bit about dietary fiber. We started talking about inulin, but there are lots of fibers. There's lots of dietary fibers, and overall fibers have been linked to positive impacts on human health. But there's a lot of variability. We can't really predict how the microbiome responds to fiber. And ultimately, this is governed at the molecular level or genetic level by polysaccharide utilization loci, which are harbored in certain gut bacteria that allow these gut bacteria to transform complex carbohydrates into kind of polysaccharide breakdown products that both the host and the microbiome can use. So we really want to understand not just different biotic factors, but also abiotic factors such as fiber, and how we can use that to tailor and control the microbiome. So this was actually, we did a study to kind of understand different polysaccharide utilization loci in B. uniformis, which is a Bacteroides species, and found kind of really interesting relationships between different genetic loci and the fibers they consume. But there's a complex set of interactions that are governing how communities respond to fiber. So Aaron Ostrom was really interested in understanding how communities respond to dietary fiber. And this has been studied in vivo with natural communities and also in vitro, but really it's not quite clear how we can predict these communities and what are the underlying ecological principles that are governing how communities respond to fibers. So we contrasted two different environments. We had the carbohydrate, which has really complex bonds, and is, can vary in terms of its physical properties, chemical properties, and has varying degrees of chemical complexity. So carbohydrates that have more diverse types of bonds are more complex. There's also different types of monosaccharides and disaccharides that comprise these carbohydrates. And so we contrasted this with the monosaccharides and disaccharides that comprise the fiber. So by having these two different conditions, we can ask, what is the contribution of glycosidic bonds to community assembly? And unfortunately, I don't have a lot of time to go into all the details. There's a lot of really interesting things we learned. But the bottom line is that these fibers, the more complex chemically the fiber is, the more robust the community is to perturbations. We also find that growing communities in complex carbohydrates limits interspecies competition, so negative interactions, and also communities are more predictable. They respond more reproducibly to perturbations in their initial abundances. So there's a lot of benefits, even at the community level, ignoring the host effects, where communities grown on fibers are actually much more stable than communities that are grown on simple carbohydrates. So this really highlights kind of some of the advantages of complex nutrients, and we're also really interested in studying this in the future in the mammalian gut as well. So we've made a lot of strides towards using machine learning models to capture microbiomes, but there are some major limitations, which is that machine learning models can make physically unrealistic predictions. So for example, they can predict negative species concentrations or kind of the spontaneous appearance of different members of the community that were never there from the beginning. And so we've actually been building on these tools further to design these tailored microbiome machine learning models that essentially require fewer parameters to generate as predictive kind of performance. And this project is led by Jaron Thompson in the lab. And so we are interested in applying this overall framework using something called Bayesian optimization, which allows us to essentially identify communities that have desired functions in a very efficient way. And we're interested in studying lots of different types of metabolites and pathogen inhibition. So this is secondary bile acids, for example, stability, fermentation end products, hydrogen sulfide, and pathogen inhibition. So we can really start to navigate a lot of these landscapes and really try to understand how we can manipulate microbiomes in predictable ways. So in the future, we're thinking about combining these dietary fibers, which have really interesting properties on the microbiome, with combinations of different bacteria that we can design to tailor and control host phenotypes and also community functions. So this is kind of a future direction for the lab. So in summary, and I actually have a second part of my talk, so I'll try to get through quickly. So in summary, microbial interactions can be exploited to design support communities that funnel flux towards desired metabolites, such as butyrate. Data-driven computational models and optimization can navigate large experimental design landscapes. Models that have really been designed and informed by in vitro data can translate to the mammalian gut. And complex carbohydrates have a lot of benefits just in terms of shaping microbial ecology. And one of them notably is enhancing community robustness. So this is allowing us to provide a foundation for thinking about how we can overcome some of these limitations of FMT and design these bacterial therapeutics in combinations with different fibers so that we can steer microbiomes to desired metabolic states. So with the remaining bit of time, I'm going to kind of touch on another project that we've been really excited about studying in the lab, which is this famous pathogen C. difficile, which I highlighted in the beginning of the talk. So C. difficile is an intestinal pathogen, and one of the most remarkable observations is that it can be eradicated very effectively with fecal microbiota transplantation. And it's one of the maybe most famous examples of how microbiomes can treat a disease. But there are a lot of limitations of FMT. We also don't really understand how FMT works. Is it a general mechanism? Is it a specific mechanism? Are there specific types of interactions that are driving this, or is it really just a general property of lots of bacteria? So we don't really understand kind of the underlying mechanisms. And also, how can we exploit this understanding for design of bacterial therapeutics? So how can we build minimal communities that are standardized, that are safe, scalable, so that we can treat diseases in a very personalized and precise way? So these projects were led by Susan Hermata in the lab, who's really kind of created a foundation for studying C. difficile, and we've learned so many interesting things that we're building on in many new ways. And so Susan was really interested in studying C. difficile using a reduced complexity community. So this is a simpler community than the 25-member consortia that I showed previously. Here we have 13 different bacteria and then C. difficile. And we chose these organisms to represent the healthy human gut, because we know healthy gut bacteria can inhibit C. difficile. We also included Clostridium scindens, which this bacteria has been shown to inhibit C. difficile in previous studies. And we used our computational experimental integration to decipher how these bacteria interact with C. difficile. This is just a computational model that we use in the lab. It's called the generalized Lotka-Volterra model. It essentially just captures the growth of individual species through its pairwise interactions alpha ij. This is the most important parameter to remember. It describes how species I impacts species J, and then it's J impacts I. And there also can be self-interactions. So we can learn these parameters from data. We give the model some data, and the model learns what the values of these parameters are, and the values tell us how the bacteria are interacting with each other. So it tells us biology. And so we did a lot of experiments to look at how C. difficile grows in the context of other gut bacteria in vitro. And we found a really striking pattern in the AIJ, so alpha IJ, which is the interspecies interaction parameter. If this is negative, that means a negative interaction between species I on species J. If it's positive, that's a positive interaction. And we see that C. difficile is inhibited by every single gut bacteria, which is a really striking and unique pattern in this inferred matrix. And we also find that C. difficile is enhancing many other gut bacteria. So this leads to a negative feedback loop on C. difficile's abundance, where C. difficile is enhancing its competitors. And we can also see this trend kind of manifest at community level if we look at the initial richness. So that's how many species we added to the community at the beginning of the experiment. And we can change the number because we have control over our community composition. And then we look at the C. difficile abundance after the community has assembled, after 48 hours of growth. And we see this negative relationship where we add more species, we get less C. difficile in the assembled community. So these transparent points are model predictions, and then the bold points are the experimental observations. But what's also really interesting here is that we have this kind of global effect of richness, but we also have these communities down here which are low richness and low C. difficile. So that shows that while you can inhibit C. diff with a high richness community, you can also design, in theory, communities that are low richness but are as effective as a high richness community just by manipulating the interactions. So a lot of variation along this y-axis. And this kind of inhibition as a function of richness is unique to C. difficile and a few other gut microbes, such as C. hirinonis and Prevotella copri. So these species are strongly inhibited when you add more species to a community. But then these other species don't seem to care very much. You add lots of species, they seem to not really mind. So their abundance does not change substantially as you increase the number of species. And so this is not a kind of universal observation. It's really specific to certain types of bacteria. And I think this provides some insight maybe into why FMT might work so effectively, is that C. diff is in this class of species that is really strongly inhibited by richness. But this may not work for all pathogens. So if we tested this with another pathogen, they might not have this same sensitivity. So I won't go into a lot of the data that Susan collected, but we found that there's a lot of different types of interactions that are all working together, and in some cases against each other, to either promote or inhibit C. difficile's growth. So we found resource competition as a mechanism, acidification of the environment, antimicrobials, and we found this one species, C. hirinonis, which is really a strong inhibitor of C. difficile. So we're interested in diving into both mechanism, but also understanding how this translates to a mammalian gut environment. So I think I'm going to maybe just mention a highlight because I'm a little running low on time here. But I just wanted to mention the kind of the findings that we have with C. difficile in this response to antibiotics. So we know that C. difficile is inhibited by lots of gut bacteria. And we also treat C. difficile very frequently with antibiotics. So vancomycin and metronidazole are two different classes of antibiotics that are used to treat C. difficile infections. Vancomycin is more narrow spectrum, whereas metronidazole is broader spectrum. They have different mechanisms of interaction. And when we use antibiotics to treat C. difficile is in the context of a microbiome. So the antibiotics are affecting not only C. difficile, but also other gut bacteria. And then we have this interaction between gut microbes and C. difficile. So how does this all kind of interplay to generate the responses we see in patients when they respond to antibiotic treatment? That's ultimately what we'd like to understand. And we started by really looking at how C. difficile responds to antibiotics in the context of other gut microbes. And Susan did a large screen to look at all the different combinations of gut bacteria with C. difficile and study how C. difficile responds either at high antibiotic concentrations or low antibiotic concentrations. And low antibiotic concentrations are physiologically relevant because at the beginning and end of dosing cycles of antibiotics, antibiotic concentrations can actually be low. And so we were interested in studying the effects in these two different regimes. And we found that while a lot of communities, we don't see an effect of the other gut bacteria on C. difficile's antibiotic response, there are a select number of communities that do alter C. difficile's antibiotic response. So it's not as simple as it might seem that you can just apply an antibiotic and you have a predictable response, but the other gut bacteria are shaping C. difficile's response to antibiotics. And just to give one example, we found there was one bacteria that had a huge effect on the minimal inhibitory concentration of C. diff. So this is just a standard dose response of C. diff in monoculture, metronidazole on the x-axis and C. diff OD on the y-axis. And as we increase metronidazole, C. difficile's growth is inhibited. And then when we add D-Piger, either it's sterile supernatant, so just sterilizing the supernatant, adding that, or in co-culture, we find that C. difficile's tolerance to metronidazole is substantially enhanced. So now it's essentially resistant to metronidazole in these concentration range. So just by co-culturing with one other gut bacteria, we can change the response of C. difficile to an antibiotic. And so we considered two different possible mechanisms. Either D-Piger is directly degrading the antibiotic, and this leads to a lower effective concentration of metronidazole. We ruled this out just by simply adding metronidazole to the supernatant of D-Piger or incubating metronidazole with D-Piger, and that did not change C. difficile's antibiotic response. So then we considered a indirect interaction where D-Piger is modifying the environment, and that in turn changes how C. difficile responds to metronidazole. And there's one really interesting property of D-Piger, which is that it produces hydrogen sulfide, and this can sequester metals in the environment, and it can turn the media black. So we knew this from the beginning. We didn't think that it would necessarily impact antibiotic response. But when we did transcriptional profiling, in the presence and absence of D-Piger, we found that D-Piger essentially creates this metal starvation transcriptional response in C. difficile. So C. difficile looks like it's metal starved when it's growing with D-Piger. And that impacts the expression levels of many enzymes that either use metals as cofactors or to acquire metal from the environment. And some of the enzymes that convert metronidazole from its inactive form to active form require some of these enzymes, or they're predicted to. And so we now had this hypothesis that C. difficile, when it's in monoculture, has lots of metals in the environment. It's able to convert metronidazole from inactive to active, and that in turn inhibits C. difficile's growth. Whereas when C. difficile is grown in co-culture with D-Piger, D-Piger sequesters these metals, and that in turn reduces the effective concentration of the active metronidazole, which then makes C. difficile resistant to metronidazole in the tested concentration range. So this is an interesting kind of example of how interspecies interactions can make a pathogen tolerant to an antibiotic, just a pairwise interaction between two species. So we're really excited to understand this more broadly across wider range of environments and understand also how this relates to clinical data. So I don't have time to talk about the last part of the talk, but I'm just going to summarize kind of this part, which is that C. difficile has many different types of interactions. There's not a single mechanism that's necessarily dominating, but resource competition, pH modification, antimicrobials, and high species richness are all contributors to C. difficile inhibition. Hydrogen sulfide production by this bacteria called D. sulfovibrio piger can render C. diff tolerant to metronidazole, and we predict this is through metal sequestration. And then these 2 points, which I didn't get a chance to talk about, but I think this really motivates the need for thinking about specific targeted therapies against pathogens, especially if we could identify more selective antibiotics to inhibit C. difficile. And we're exploring now lots of different strains, clinical isolates of C. diff. C. diff has an enormous genomic variation, more so than a lot of other bacteria. And so we're trying to understand how this genomic variation impacts interactions in the gut and how we can use this to design robust and minimal C. difficile, anti-C. difficile communities. So I didn't talk about a lot of the other work that's ongoing in our lab, but we are looking at different applications of microbiome engineering and bioprocessing and in agriculture and human health. Human health is our kind of maybe major focus. And we use a wide range of different types of computational models to really understand microbiomes at a more mechanistic level and predict and design their properties. We're also looking at host microbe interactions and using ultra high throughput approaches to study microbiomes and their interactions. And we also are really interested in synthetic biology, so engineering next generation therapeutics with design metabolic pathways and biosensors that can sense the environment and be embedded in other communities that have beneficial functions. So with that, I'd like to thank our group and collaborators. We've had really fantastic collaborators both here at UW-Madison and other universities as well, like USC and University of Michigan, and our funding, and I'm happy to take any questions.
Question for Ophelia. I think I should use the microphone. Pass the microphone around. It's being recorded.
Amazing, obviously. But I had one question, which is like you kind of presented. In some cases, there would be these like keystone species where it's like the presence or absence could be like a strong predictor of butyrate, like being made or not. And in other cases, there was this concept of the community support community that was kind of like a booster of that. And I was curious, as you've looked at more and more systems, is it more do you sort of feel like one of those paradigms is more dominant for kind of controlling metabolic flux, or is it more like, both, it just totally depends on kind of the details of the metabolites or what's being made?
Yeah, that's a great question. I would say that like the keystone species we've found are pretty rare in the community. There's a lot of metabolic redundancy, and so many species can actually perform similar functions, but then there's these unique species that have metabolic roles that no other species has. And that's really what defines a keystone species. It's contributing in a unique way, an important way to a community function. So yeah, I think that when we talk about really complex communities, it's going to be difficult to find keystone species, and it really also depends on the function. So as you add more redundancy, there's likely many different types of interactions that have to be changed to really alter a community function. But if you were looking at, say, for example, some very unique function, like production of one specific bile acid, that's very unique, then there may be keystone species for that because there's only a few species that really can produce it. So it depends on the type of function and the richness. Yeah.
How does hydrogen sulfide inhibit butyrate production, and does butyrate affect the growth of C. difficile?
Yeah, so hydrogen sulfide's impacts on butyrate is something we're investigating now. I don't think I have a good answer to it. It may have something to do with metal sequestration too, but we don't know yet. There are a few different hypotheses that we're testing now. And so that I think is really going to be interesting because there's this negative relationship between hydrogen sulfide and butyrate production, especially in certain disease states. It's not necessarily causal, but there's correlations there where hydrogen sulfide is really high and butyrate production is low. So it'll be interesting to know like what those mechanisms are and whether they generalize to like clinical data potentially and in vivo. And then the second question was about hydrogen sulfide on C. difficile's growth.
Is it mediated by its effect on butyrate?
So butyrate, we've shown and others have shown that butyrate inhibits C. difficile's growth. And that's true across many butyrate producers. Other butyrate producers are also inhibited by butyrate. Hydrogen sulfide does not, as far as I remember, does not have a substantial effect on the growth of C. difficile directly. And I guess if it's inhibiting butyrate production, it could, in theory, enhance C. difficile's growth by limiting butyrate production. But yeah, we haven't explored that directly. Could be interesting.
Great talk.
Thanks.
So there are lots of other ways to sequester metals than spitting out hydrogen sulfide. Some of them are even produced by bacteria. Is that involved in anything here? Have you looked at that?
So you were talking about siderophores or?
Yeah.
So metal availability in the gut is something we haven't really looked in depth at yet. My understanding is that like iron and other metals might be limiting in the gut. They're also very important for like pathogen virulence and C. difficile too as well. But yeah, I think, so we haven't looked into that. I think that could be really interesting to explore in the future. See if there are other mechanisms that it could sequester metals and their effects on the gut. Yeah. Oh, sure.
Because bacteria produce some, acquire their own metals under very difficult circumstances.
Mm-hmm. Yeah.
Yeah, no, that was an absolutely impressive body of work, Ophelia. Congratulations. But I was really curious about when you looked at the fiber and the impact of the glycolytic bonds, you saw that that limits interspecies competition. And I'm curious if you can speak to the mechanism. Is it like that the fiber creates like a unique ecological niche, or is it potentially like a substrate competition, or what do you think is driving that?
Yeah, and it's even simpler than that. We think that it's actually just growth restriction, and it has to be a very specific type of growth restriction. So I don't think if you restricted the growth of communities with like a wide range of mechanisms that you'd necessarily see the same effects that we're seeing. But growth restriction by these complex carbohydrates seems to reduce the amount of modification that the bacteria have on their environment, which then reduces interactions and then promotes the stability. It allows certain types of taxa that are what we call non-responders, so they're not growing in response directly to the fiber. They're growing on other nutrients, so other orthogonal niches. So that allows those bacteria to expand when you restrict the growth of the community with complex carbohydrates. And that in turn stabilizes the community. So it's really kind of a growth restriction effect is our current hypothesis. Yeah.
So Ophelia, so first of all, let me just say that I am always overwhelmed by the complexity when I hear about microbiomes, but I think it's super cool the way you're approaching it, and I feel like you really are starting to get a handle on at least manipulating the complexity, which I find very exciting. So the question is, so in our natural microbiomes, you say we have about 100 species, right?
Mm-hmm. Roughly. Mm-hmm.
And but if you are introducing new microbiomes and engineered microbiomes, which seems to be the direction you're going, what is the optimal number of different species for maintaining a staple microbiome? If you introduce 20, will it go up to 100 quickly, or is 5 enough? What is the optimal number, or is that a crazy concept?
So optimal number for what function? Just for colonizing and maintaining stability?
Colonizing in a healthy microbiome. I mean,
yeah, so that, as far as I know, has not been looked at directly to see if there's an effective number. So I think, richness has these kind of systems level effects. So as we change richness, We get certain types of like changes in the statistical properties of these systems. But if you look at an individual community basis, there's a huge variation. So for two species community, we could have like super high butyrate, or we could have very low butyrate, or we could have very high C. diff or very low C. diff. So I think there's these like trends with richness that are kind of statistical. But if you look at an individual community, I don't think richness is going to be a great predictor, would be my guess. But I don't think anyone's looked at that particular question. What they have looked at is like how different strains or taxa can engraft into patients following FMT. And so that's something that we're interested in also looking into with clinical data, because I think that could provide some insight into possible interactions that influence C. difficile's ability to be eliminated with FMT. But as far as I know, no one has done the simple experiment of changing richness in a mouse, with C. diff. Like, that's the experiment we did in vitro. No one's done it in vivo. And I think our results point to the fact that you need to control for that overall, because it can have very large effects on kind of the conclusions that we draw. So yeah, I don't have an answer to the question, but I think richness is not a predictor of quantitative behavior. It's going to predict maybe some statistical effects of either like increasing or decreasing globally, but there's much more work that's needed. Yeah.
So Ophelia, I wanted to ask a question about the negative feedback loop you were describing with C. diff and other bacteria, because it seems like, in principle, that predicts C. diff can never take over. But something must undo that in the case of a C. diff infection and disease state.
Yeah.
So do you have some speculation about what happens that undoes the negative feedback loop?
Oxygen is a major factor. We haven't actually tested this, but in the inflamed gut, oxygen increases, and that will in turn inhibit good bacteria that are playing beneficial roles. And the causality of these things is really not known, so this is just all speculation. But I think oxygen plays probably a big role in allowing certain types of microbes to exploit the inflamed gut. And C. diff is actually quite sensitive to oxygen, but it's not as sensitive as people think. So I think that might be one major factor, but yeah, it's a great question. I think the inflamed gut has different types of resources than the gut when it's in homeostasis. So pathogens know how to exploit those nutrients as well. So that's another factor. Yeah.
Any more questions? Can you say in just 1 minute what the secret sauce is to teaching undergraduates to do this kind of modeling and to really accomplish this stuff at a very high level?
I'd say it's still a work in progress, but I think so. We start with a lot of basics in our class, and really starting to kind of build an intuition with lots of examples and things that they might be familiar with in chemical kinetics. And a lot of the math that we teach in the class is very easy to learn. So we can just teach them a few simple rules, and they can learn those concepts without having to understand the depth of math as a field. And then also, I think there's some like tools in Python that have been really in coding that have been really powerful for just like simulating a model. So you don't actually have to understand every little detail, but you can just simulate and analyze these models with computational techniques. But yeah, I think it's still a challenge. Like, we're still trying to figure out how to balance, because there's people that come in with students that have like much more computational background. They might be engineering majors, and so they have a lot of expertise, and then there's students that have never seen a differential equation before, and trying to kind of figure out that balance is still a work in progress, but it's been really fun, and I think I've learned a lot, so.
Very impressive. Thank you. Wonderful seminar.
Thanks. It was longer than I thought it was going to be, but I didn't practice it, so I didn't know how long it was going to be. Thanks. Thanks.